What do you need to know from all this protein synthesis? First, protein synthesis is carried by RNA molecules. Instructions comes from DNA molecules, from the gene. So what is the genome? The genome is the entire DNA molecule with all the genes in the cell. Gene is only a portion of the DNA that carry information or instruction for only one protein. Next thing you need to know is transcription. The term transcription. Transcription means formation of mRNA molecule from the DNA molecule, and it takes place inside the nucleus. Then the mRNA leaves the cell, the nucleus. The mRNA molecule is divided into 3 nucleotides pieces. Each 3 nucleotide or triplet of nucleotide is called codon. So if I ask you the codon is part of, you will say the mRNA. How many nucleotides the codon is made of? Only 3, and they represent only 1 amino acid. What about the genetic code? That's the codon. It's part of the mRNA and it's only 3 nucleotides. What about the genetic code? The genetic code is the sequence of nucleotide on the mRNA. The genetic code is a sequence of nucleotide on the DNA, and it represents the sequence of amino acid in the specific protein. So the genetic code is long. It represents all the amino acids of one specific protein. The codon, only triplet of nucleotide and 1 amino acid. The mRNA, once it reaches the ribosomes, the next term that you need to know is translation. So translation takes place at the ribosomes, where in the cytoplasm, different than transcription that takes place in the nucleus. What is the definition of translation? Translation means the formation of a polypeptide chain or protein from the messenger RNA with the help of tRNA that brings those amino acids. So translation is the formation of a polypeptide chain from the messenger RNA. Where does it take place? In the ribosomes within the cytoplasm, whether free ribosomes or on the rough ER. That takes us to the transfer RNA, which are these. The transfer RNA is an RNA molecule that has specific nucleotide sequence that is made of only also 3 nucleotides that represent the amino acid that this transfer RNA can carry. So it matches the amino acid that it carries. What is this triplet of nucleotide called? It's called the anticodon. So if I ask you what is the anticodon is a specific triplet of nucleotide on the transfer RNA, this part. Why is it called anticodon? Because it's complementary to the codon of the mRNA. Complementary to the codon on the mRNA. Okay. And this is basically what I was going over. Messenger RNA. It's inside in the nucleus as a complementary sequence to the DNA, which is a template, and this is called transcription. The codon is a triplet of nucleotide in the messenger RNA. So each 3 represent 1 amino acid. That's the codon. Messenger RNA leaves the nucleus associated with ribosomes in the cytoplasm or on the ER, where protein synthesis takes place, called translation. So translation is the formation of protein from a messenger RNA with the help of transfer RNA. What does the transfer RNA do? The transfer RNA carry amino acids, or one amino acid. It has a specific triplet of nucleotides called the anticodon that is complementary to the nucleotide on the messenger RNA, and again it represents one amino acid. Excuse me. Ribosomal RNA, just know that it's part of the ribosomes, whether free or on the ER. Its function is to hold the messenger RNA in place and start reading the message. Ribosomes will contain enzymes that help join the amino acids together to form the peptide bond and hence the polypeptide chain. And at the very end, again, the polypeptide chain is released to become the specific protein. This way we finish chapter 4. I'm just going to go over the last few questions that we didn't answer, and the ones that we did. Let's just go over the gene and the genome. The gene is a part of the DNA, represent only one protein. The genome is the entire DNA molecule in the cell. DNA information provides instruction for the cell to synthesize protein molecules. So the answer is D. Why DNA replication is essential? Because DNA replication provides each new daughter cell with an exact copy of the genetic information that is necessary for the cell to carry its different metabolic reactions. Describe events of DNA replication. DNA molecule split, and as it's splitting, each strand of the DNA is used as a template to bring complementary bases and start to form a new polypeptide chain. At the end of DNA replication, each of the two DNA molecules will be made of one original or parental DNA strand and one newly formed polypeptide chain. So the events are splitting, new nucleotides being added complementary to the parental strand, and then at the end we will have two double-stranded helix, each with a parental strand and a newly formed strand. Question 21. If a strand of DNA has a sequence of such, then the sequence of an mRNA molecule transcribed from it is. Now, transcription means again formation of mRNA from DNA, and it takes place in the nucleus. Now, in order for me to use this sequence to form mRNA, notice that RNA molecule will have uracil instead of thymine. So in front of the A, I will put U. In front of the T, I will put A, C, G, C, U, A, G, G, C, G. Now, if I change this question and I put, if a strand of DNA has a sequence of such, then the sequence of the complementary DNA molecule replicated from it will be, then I will use a thymine. I'm not going to use a uracil, so I will put T, A, C, G, C, T, A, G, G, C, G. Okay, so the trick will always be in the A and the T. 22. Distinguish between transcription and translation. Transcription, again, the formation of mRNA molecule from a DNA molecule, and it takes place in the nucleus. Translation is the formation of polypeptide chain or amino acid chain from the messenger RNA and it takes place in the ribosomes in the cytoplasm of the cell. Uh, 23 describe function of ribosome in protein synthesis. Ribosomes fix the mRNA in place and read the message from the RNA, and this is where translation takes place. 24, calculate the number of amino acids encoded by a DNA sequence of 27 nucleotides. Now, before you answer, think. The question is giving you the number of the nucleotides, which is a bigger number. So I have to divide by 3. So 27 divided by 3, then this amino acid chain will be 9. If the question is telling you calculate the number of nucleotides needed to synthesize an amino acid chain of 9 amino acids, or a protein chain of 9 amino acids, then if the number that I'm giving you is for amino acid, then I will multiply by 3. So the trick is always which number I'm giving you, the bigger or the smaller. The bigger is the nucleotide, then I will divide by 3 to get the smaller one, or the shorter one. So a 27 nucleotide chain gives a 9 amino acid chain. Last thing is gene expression. Gene expression is basically how each cell chooses to synthesize or using a portion of the DNA molecule to synthesize specific protein and not using other portions. And basically gene expression shows how cells can differentiate from others. So it is, gene expression is how cells choose to turn on some genes and turn off other portions of the DNA, and this helps in differentiations of the cells so that each cell will carry a different function, because not all cells will express all the DNA information inside it. And that's what is gene expression, what gene expression means. And we are done with chapter 4.